All right, good morning, everybody. Looks like we're after 9 o'clock, so we'll go ahead and get started. On behalf of Washington Association of Wheat Growers Ammo Program, I'd like to welcome you to the 2021 Wheat College. It has a little bit of a different look this year, but we're glad so many of you could join us. For anyone not familiar with our ammo program, it's the grower educational arm that has offered seminars and more since 2009. We thank the sponsors that make this program possible. For today's webinar, please use the chat box feature to ask questions throughout the presentations. To guarantee your pesticide credits, we will ask you to enter your license number in that chat box, probably at the break when we move on to our next speakers. Feel free to make this an interactive event and jump in, but otherwise, please keep your line muted. There will also be time at the end for questions and discussion. At the close of the webinar, we will draw for a winning participant to win a Blackstone grill from WAG and other giveaways. Today's featured speakers are from Washington State University Extension. Each will introduce themselves as they begin their presentation, and we will start with Drew Lyon. So take it away, Drew.
All right, thank you, Lori. Let me get my screen shared here. Can everybody see that? Yep, all right.
Good.
So let's see. Let me pick a tool here to help me. There we go. All right. My talk today is on managing herbicide resistance in wheat production systems. I hope to talk a little bit about how herbicide resistance starts, some of the things we can do to help slow it down. If we use herbicides, we can't really stop it, but we can slow the process down and maintain effective herbicides longer than we might otherwise. And then I'm going to talk a little bit about some of the research I've been conducting over the last few years on the topic of herbicide resistance and some of the management strategies, particularly herbicide management strategies we can use to help us manage our herbicide resistance situation. So I start my discussions on herbicide resistance with this gentleman, Charles Darwin. I think most people are familiar with the gentleman. Over 150 years ago, he published a book that kind of revolutionized biological sciences. When I asked my audiences, my live audiences, the name of the book, the typical answer I get is on the origin of species. And that is only a partially correct answer, because the real title of the book was On the Origin of Species by Means of Natural Selection, or the Preservation of Favored Races in the Struggle for Life. So this part of the title, Origin of Species, is what a lot of people remember and probably get a lot of discussions going around dinner tables and across bar stools. But really the real discovery, the thing that really profoundly changed biological sciences, was this part: natural selection, the process by which species evolve and change. It wasn't so much this that gets all the attention, it's this. And there's very, very little disagreement on the process of natural selection in the scientific world. It is probably one of the strongest supported scientific theories there is, even stronger than gravity. It is a very strong idea. You can watch it happen in petri dishes. You can watch it happen in jars with fruit flies. It's the process by which species evolve, and for which Charles Darwin is held in high esteem in the scientific community. In order for natural selection to occur, three things have to be true. If you don't have these three things, natural selection doesn't occur. So there has to be a struggle for existence. Life can't be easy. We can't all be laying around in our cots and eating bonbons, or we don't get natural selection. And I think if you talk to most people, they'll tell you life can be a struggle at times. So you have to have a struggle for existence, and you have to have variation in the population. If every individual in the population is exactly the same as every other individual, there cannot be natural selection. And then that variation has to be inheritable. So parents have to be able to pass that variation on to their children and their children onto their children. If you have all three of these things occurring, then you can have natural selection and species can evolve and change over time. This is a picture I took back around 2014 or 15. One of my first experimental plots up north of Davenport, Washington. This is a spring barley field. I've gone out and sprayed various treatments in here, and this barley field was infested with mayweed chamomile, what many growers call dog fennel. And the product I sprayed in here was an ALS inhibiting herbicide. I don't remember specifically which one, but it was a group 2 ALS inhibitor. And if you look here, what you can see is that the population of mayweed is segregating for resistance to that herbicide. So we have some plants that are dead, some plants that are looking pretty sick, and some plants that don't look like they're affected at all. So if we fast forward this picture, say a month or two down the road, it's about harvest time. Which one of these plants are going to have grown larger and produced more seed? It's going to be plants like this. These aren't going to produce any. These are going to produce a few, and these are going to produce a lot. So the next year, when I come back to this field, if I apply the same herbicide, more of the plants are going to be like this than like this or like this. And if I keep putting that same herbicide on year after year, pretty soon the entire population, or almost the entire population, will look like this. And that's basically how we are. I guess it's the principle of natural selection. You can call it artificial selection because humans are putting the selection pressure on the population with the herbicide. But that's how we drive a population from being susceptible to being resistant. And this is a kind of a case study on how that happens. Now I could stop here because that pretty much explains it, but then you don't get any pesticide credit. So I have to talk a little bit longer, and we'll go through here and give a little fuller explanation. So this is basically a schematic of how weed resistance progresses. This schematic, this example is actually taken from the state of Washington. This is exactly what happened with a product called Glean. And those of you who might not have as much hair on the head as some others might remember Glean. It was introduced in the early 80s. It was the first group 2 herbicide, the first ALS inhibiting herbicide introduced into the wheat market. And it was dynamite. It controlled pretty much every broadleaf weed we had out there, and growers really liked it and used it consistently, and herbicide resistance developed quickly. And so this example is from Russian thistle and prickly lettuce, both that developed resistance to Glean very quickly. Now, in this example, this is day 0, year 0. We've not applied Glean to this population at all. And what we notice is that even though no herbicide's been applied, there's still a small percentage of plants that are resistant. 0.0001 plants. That's one plant in a million. This is a mutation. Mutations happen all the time. As our DNA gets replicated, there's a mistake, and that mistake gets replicated, and you have a mutation. If that mutation is positive, it tends to increase in the population. If it's negative, it tends to decrease in the population. And if it's neither positive or negative, it tends to exist in the population at a very low frequency. And it is estimated in the case of resistance to Glean herbicide in Russian thistle, that mutation existed at about one plant in a million. So so the herbicide does not cause the mutation. The mutation is naturally occurring out there, but it's neither a positive or a negative mutation. So it exists low. So 1 in a million, never applied. We go out and we apply our herbicide the first time, and we get excellent control. We get 99.9% control. In fact, our population we've killed off 99.9% of the weeds, of the susceptibles, but we've increased now the percentage of resistant weeds from 1 in a million to about 1.5 per 100,000 plants out there. Still a low number, but we're starting now to increase the soil seed bank of the resistant type. But that herbicide worked really well. So we're going to go out and we're going to apply it again, because when we find something that works, we tend to stick with it. So we applied a second time, and we get 99.98% control. We've now moved our population from 1.5 plants per 100,000 to 2 plants per 10,000 with resistance out there. And again, we're starting to put a little more resistant seed into the soil, but control is still excellent. We still, we don't really see that small change in resistance. And so it's a product that works well. We go out and apply the third time. And Glean was a product that you could apply both in the wheat and the fallow at that time. You can now only apply it in wheat, but it could be applied in wheat and fallow. So that's what was done. And Glean has a long soil residual, so it exerts the selection pressure for a long time on the population. So we apply it a third time, and we still get excellent control. We now got 99.8% control. We've now enriched our population to about 3 in 1,000 plants. And again, you can see we're continually filling up the seed bank with resistance, but we're still getting excellent control. So we applied a fourth time. Now this fourth time, we get about 96% control. Still excellent control, but we noticed it this year. We noticed that, it's not quite as good as it's been before, but maybe the wind was blowing, or I didn't quite get the mix right, or, I skipped some areas or something, but it's still good. And we're going to go out and apply it again. And you can see now our seed bank is really starting to fill up and with resistant seed. And we applied a fifth time, and whoa, now more than half our plants are resistant to our Glean. And now we say we have a failure. We've been building that failure up all along, but it's not until this fifth application that we noticed that it failed. And at this point now, our seed bank is full of resistant seed. And unless there's a fitness penalty for this resistance, which in the case of Glean, there's not, even if you stop using Glean, your population is going to stay resistant for a very, very, very long time. A resistance penalty or a fitness penalty is when in the absence of the herbicide, the susceptibles produce more seed than the resistance ones. But if in the absence of the herbicide, both the resistant and the susceptible produce the same amount of seed, then you're not going to go back. And with most sulfonylurea herbicides, there is no fitness penalty. It's a single 1 gene mutation, and it doesn't seem to affect the plant's ability to reproduce. Now, in some triazine herbicides, they do see a fitness penalty. So in those, you could stop using it for 10 years or so, and your susceptibles would slowly take over, and you could go back to using it. But that won't be the case for many of our herbicides, particularly the ALS herbicides. So this is exactly what happened with Glean in Washington State in relation to both Russian thistle and prickly lettuce. It's about as fast as we've seen herbicide resistance develop, but it is possible if you keep applying the same strong selection pressure on a population, and there is a resistant biotype out there that can survive that application. So how long does it take? It depends on a number of factors. The cultural practices being used, the frequency of herbicide use, what the herbicide mechanism of action is. The ALS inhibitors, a single mutation can result in herbicide resistance. So in the case of ALS and ACCase inhibitors, so group 1 and group 2s, that resistance has happened rather quickly. But in cases like 2,4-D or glyphosate, it's taken quite a while. If you remember back, Monsanto used to say that weeds would never develop resistance to glyphosate to Roundup because it's a very complex action mechanism that the plant has to overcome. A single mutation is unlikely to do it. And it did take a long time to develop herbicide resistance to the glyphosate, but we now use it. We use so much of it in so many situations that we have started to develop resistance to that too. So these factors come in play, and then the biology of the weed species. Cross-pollinated, highly diverse species tend to develop resistance more often because there's more genetic diversity to select from. And the frequency of resistant biotypes among the species. Is it one plant in a million, one plant in 10 million? Maybe there's no mutation out there at all, in which case you wouldn't select for resistance. So you put all these factors together, and that dictates whether it takes 5 years, 10 years, 20 years. But eventually, weed populations will develop resistance to herbicides given enough time and enough use. Here's a global increase in unique resistant cases. You can see the chart starts back in the mid-50s. 2,4-D was introduced into the marketplace after World War II as a product developed during World War II as a weapon. And then it was released commercially in the 1950s. And shortly after its introduction, we had a couple species develop resistance to 2,4-D. So herbicide resistance has been around as long as we've had synthetic organic herbicides. It's not something new. It's just you can see that the pace of increase has really accelerated. Some of that's the introduction of ALS inhibitors, ACCase inhibitors that develop resistance fairly quickly. But it's also the case that we've relied more on herbicides in the last 20, 30 years than we did before. We used to use more methods of weed control than we did back in the 50s, 60s, and 70s. And so herbicide resistance is developing more quickly. Here's a slide that shows it by mechanism of action. So here are our triazine inhibitors, things like atrazine, simazine. When I was a graduate student in the 80s, this is what most of my fellow graduate students were working on, was triazine resistance in Nebraska in corn. Here's the one that should worry most wheat growers. This is the ALS inhibitors, the group 2s. It's the mechanism of action used most in wheat. And you can see from their introduction in the early 80s how quickly herbicide resistance has developed in those. Here's the ACCase inhibitors, the group 1s, another common mode of action used, or mechanism of action used in wheat. Here's the one that's gaining a lot of attention in the news. This is group 9. These are the glycine inhibitors. This is glyphosate. And you can see glyphosate was introduced in the 70s, and we didn't develop resistance until the mid-90s. And you may remember what happened in the mid-90s. In 1996, Roundup Ready soybeans were introduced into the market. And in 1997, Roundup resistant Roundup Ready corn and cotton were introduced in the market. And then we started spraying our weeds, a lot more acres, a lot more weeds. And instead of just spraying before planting and after harvest, we could spray before planting, a couple times during crop growth and after harvest. So the selection pressure really went up, and given enough selection pressure and time, now we're developing a lot of resistance to glyphosate in our systems. Here's increase in unique resistance cases in the USA. So breaking out the worldwide to just the US, same sort of curve as we saw for global cases. If we look at it by crop, until the mid-1990s, corn had the most cases, but after the mid-1990s, you can see that wheat's taken over as having the most cases of unique herbicide resistance. The other troubling piece of information is that we're developing more weeds with resistance to more than one site of action. So this shows the number of weeds that have resistance to 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different mechanisms of action. So we have some weeds that we're pretty much running out of the ability to control. In our area, Italian ryegrass is one of these. We have Italian ryegrass resistant to pretty much every mechanism of action we have. There's still a couple that are working, but if we put too much pressure on those, they'll stop working too. And this is resistant biotypes where just the biotype has multiple resistance. So the other one was across, you know, the weed species and all biotypes. This is just a single biotype with 2, 3, 4, 5, 6, 7 different mechanisms. So one weed biotype in one field that you can't control with any of these herbicides. So that's kind of a scary situation when you start developing weeds that have multiple mechanisms of action that they're resistant to. Here's some of the species that are the winners. Lolium rigidum. This is rigid ryegrass. This is the problem in Australia. When you hear that the Australians have a heck of a problem controlling weeds, this is one of them that they really struggle with. Basically, they've run out of herbicides that they can control rigid ryegrass with. Herbicides just aren't the way to do it anymore. We have annual bluegrass here. Here's our Italian ryegrass.
And there's poles that
we have Italian ryegrass here at 8, and here's wild oat. So we have a few species that are doing quite well in developing resistance to the mechanisms of action that we have. So the current status of resistance: there are no herbicides with new mechanisms of action in advanced development trials. Now this statement's maybe a little old. DuPont, before they merged with Dow, had a new mechanism of action. They sold it off as part of the merger to form Corteva to FMC. So FMC has a new molecule. I don't know of any university researchers who have seen it. And my information from FMC suggests that if it makes all the hurdles and gets commercialized, it will be commercialized in corn and or soybean before it's commercialized in wheat. But there may be a new single mechanism of action coming somewhere in the next 5 to 10 years. So this part's changed a little bit, but it's still not very promising. The last new mechanism of action was introduced over 30 years ago. So it's been more than 30 years since we've had a new mechanism of action. And if this next one comes out, a new mechanism every 30, 40 years doesn't bode well for fighting herbicide resistance. This chart helps to explain at least why we haven't seen, partially explain why maybe we haven't seen any new mechanisms of action lately. A bit complicated slide. This is the US patents for new herbicides here with the dots. And this is a cumulative count of new active ingredients into the marketplace. So you see back prior to 1996 and the introduction of glyphosate or Roundup Ready crops, you have to have a patent before you can have a new AI. Patents are going along at a nice clip here. New AIs are coming out at a pretty regular basis. And then in 1996, we kind of peak and we've fallen off a cliff since then. And we don't get as many new introductions of new active ingredients. The introduction of Roundup Ready crops in 1996 sucked all the profitability out of the herbicide market in the US. Basically, companies make money by selling herbicides in the corn and soybean market. And if they can't make a profit in the corn and soybean market, they don't make a profit in the US. And they can't come out with things in wheat and other stuff. So when Roundup Ready soybeans came out, everybody was cheap, easy, effective, and people no longer bought products. I was a technical service rep in the late 80s for American Cyanamid, and we came out with the imidazolone herbicides. Pursuit, Scepter, charged a lot of money for them, made a lot of money selling them to soybean growers, and in 1996, they all switched to Roundup Ready soybeans. American Cyanamid sold out to BASF in the late 90s or early 2000s, and so did a lot of other companies. So all their discovery programs went away. Only the big players still had discovery, and they didn't really see herbicides as the place to make money. So if you've noticed, there's been a fair number of new insecticides and fungicides, but not very much in the herbicide market. So that might be starting to change now that we got Roundup or glyphosate resistant weeds. But we're never going to be back, in my opinion, to the heyday of all these new products coming out for herbicides. We have to work with what we have today. We lose the effective products we have today. There's not much coming down the road to save us. So, number of weeds with herbicide resistance continues to increase, and the number of weeds with herbicide resistance to more than one herbicide continues to increase. So this is not a great scenario for maintaining good control with herbicides alone. So how do we avoid or delay herbicide resistance in weeds? We have to use more integrated weed management. So if you take Weed Science 101 here at Washington State University or anywhere, you'll learn that there are 5 basically approaches to controlling weeds: sanitation, mechanical, biological, cultural, and chemical. If you're in an annual cropping system, there aren't very many known successful biological control methods out there. So there are in perennial systems, but not so much in annual systems. So now you're down to 4 approaches that you need to work. But what happens if you're a direct seeder or a no-tiller? You can't use mechanical. So now you have to rely on these 3. And what a lot of direct seeders rely on is chemistry. They've replaced tillage with chemicals. But I've just shown you that our chemicals are becoming less and less effective, and we need to spend more time on the cultural and sanitation, things like crop rotation and some of the other approaches we have. Now, organic growers, they give up the chemical. They tend to substitute mechanical for chemical. So anytime you give up something, you have to really step up your game. And what the organic producers tend to do is they really have some very sophisticated crop rotations to help them manage both weeds and nitrogen. And I don't see the no-tillers, well, there are some. A lot of no-tillers just kept the same rotation they used when they had tillage, and they changed to no-till. And I would argue that if you change from tillage to no-tillage, you need to change your cultural practices to help stay on top of the weeds and really up your game in that area. So, general principles of integrated weed management: use agronomic practices that limit the introduction and spread of weeds. So prevent weed problems before they start. Help the crop compete with weeds. That is, help it choke out weeds. And we're lucky, winter wheat is a very competitive crop. So if you can do everything you can to grow a good competitive wheat crop, you've gone a long way to controlling weeds. I've put out a lot of weed control trials in my career, and herbicides always look better in a good stand of wheat than they look in a poor stand of wheat. And then use practices that keep weeds off balance. So don't allow weeds to adapt. And that basically means don't keep doing the same thing over and over and over again. Because if you do, you'll find those weed species or weed biotypes that thrive in that system, and they'll become your dominant weed. So every once in a while, change things up: a new crop, a new timing of herbicide application, change in herbicides, maybe introduce a different tillage system every now and again, but don't stick with the same thing over and over again. This is probably one of the toughest things to do, because as humans, when we find something that works for us, we like to stick with it until it doesn't work for us anymore. But if you do, weeds resistance is likely to come. This is a nice table to look at the risk of resistance on a per species basis. So whether you're at low, moderate, or high risk for developing herbicide resistance, and then the different management options. If you use a herbicide mix or rotation in cropping systems, you use more than 2 modes of action, you're at low risk. You only use a single mode of action, you're at high risk. If you use cultural, mechanical, and chemical control, you're at low risk. If you rely solely on chemicals, you're at high risk. Here's one that the Australians are really focusing on: weed infestation. If you have low weed numbers, you're at low risk. If you have high weed numbers, you're at high risk. So try to keep your weed numbers as low as possible. The reason for that is it's a numbers game, right? If one plant in a million has that mutation that confers resistance, if you have less than a million plants out there, the chances of you having that resistance are lower than if you have 10 million plants out there. So keep weed numbers low and your chances of developing resistance are also low. We recently, I with several of my colleagues in the Pacific Northwest published an extension guide on managing herbicide resistance. So a lot of these details can be found in this publication, PNW 754, which is available on smallgrains.wsu.edu. So let's talk a little bit about some of the research I've done to try and deal with this. This is a field in western Whitman County in 2015. The grower sprayed a quart of RT3 on here and did a nice job of controlling everything except this weed here. And if you look closely, you can see there's a trail of Russian thistle here. This is one resistant mother plant that blew across the field and spread its seed in that path. And that's a very good indication for a tumbleweed anyhow, like Russian thistle, that you have a resistance issue on your hands. We went out and collected some of those plants and collected seed off of them. And I had a graduate student at the time, John Spring, who conducted a dose response study in the greenhouse where he's applied different rates of glyphosate to both the susceptible and the resistant seed. And he found that the resistant seed was 4 times more resistant to glyphosate than the susceptible. So in the greenhouse, it took 4 times as much glyphosate to get the same level of control as we got with the susceptible. He took it a step further and went out to land and did this in Pullman as well. And he planted the resistant and the susceptibles, and then he sprayed them with various rates of glyphosate. This is the highest rate he used, 3 pounds of AE of RT3. So this is a gallon of RT3. And you can see the susceptible is toast. The resistant one may have the sniffles, but it really isn't hurt. We estimated it would take 2 gallons of glyphosate of RT3 to control that. And at that point, it becomes very uneconomical to be using glyphosate to control Russian thistle. So Russian thistle resistance to glyphosate was also reported at the same time in Northeast Oregon and in Eastern Montana. So it's developing around the Western US. It's a tumbleweed, so it's going to spread just like kochia did in the Great Plains. And I'm sure there's more glyphosate resistant Russian thistle in Eastern Washington now than there was in 2015. So it's something we need to deal with. One of the ways they've been trying to deal with glyphosate resistant kochia in the Great Plains is by using soil applied herbicides in their fallow. So we wanted to do a study to see if we could do a similar thing with Russian thistle. So we had 3 treatments. We had Spartan Charge, which is sulfentrazone plus carfentrazone at 8 fluid ounces of product per acre. Flumioxazin plus pyroxasulfone, which is fierce, at 4.5 ounces per acre. And metribuzin, we use Tricor 75DF at 10.5 fluid ounces per acre. We applied it either in late fall, so late November, early December, late winter, which would be late February or early March, or we split it, half the rate in the fall and half the rate in the late winter. And we had several sites. I cooperated with Judith Barrasso down in Pendleton with the Oregon State University. She put out the trials in Pendleton in 2018 and 19 and in Morrow in 2019. We put out the study in Lind in 2019 and the Horse Heaven Hills in 2019 and Ralston in 2020. The bolded text shows the sites where we had pretty good Russian thistle densities, and I'll show you the data from those sites. We collected other weed issues or data from some of these other sites, but we're going to focus on Pendleton in 2018, Lind in 2019, and Ralston in 2020. We took weed counts monthly starting in May, and then we spray out the plots to kill out any weeds that were up at the time. And we ran this just talks about the stats. We did a randomized complete block design. Here's a picture from Lind in July 2019. This is our non-treated check here, so you get a sense for the density of the Russian thistle. Oops, and here is the sulfentrazone plus carfentrazone. So the Spartan charge here that was applied in the late fall. So in July, it was still very clean compared to the check. So looking at the data, this is from Pendleton. This is our wettest site. So they got the most rainfall here, and it did seem to affect some of the treatment. So here's our check with over 15 Russian thistle plants per square meter, similar to what we found where we put metribuzin on in the fall. So the metribuzin, especially at Pendleton in the fall application, just didn't last long enough to provide us good control of Russian thistle into May. This is the May data. And the Spartan charge in the fall broke down a little bit, still better than the check or the metribuzin in the fall, but a little breakdown. But the winter application or the split applications of the Spartan charge look good. All the applications of the fierce treatment look good. And the metribuzin, if we applied it in the late winter or split applied it, wasn't too bad. So those are May counts. Here's the May and June counts. They didn't get any more emergence after June. Actually, this is June, July, and August. They did. I don't know why that's not showing up. Anyhow, this is for the entire summer period. You can see we got a bit of addition after the May, but most of our population came up in May. And very similar to what we saw previously. Our check and our late fall applications of metribuzin were not very effective. Our Spartan charge in the fall broke down a bit, but our other application times weren't too bad, especially the late winter of Spartan charge, the split application of fierce, and the winter application of metribuzin. Here's the Lind data. Lind's a drier site, and so at Lind we didn't see the breakdown of the fall application of Spartan charge. The fall applications, late fall applications worked as well as the late winter or the split applications here. The fall application, late fall application of metribuzin, better than it was at Pendleton, but drops off a bit, not as good as any of these other treatments. If we take a look at it throughout the rest of the summer, you can see that the Spartan charge treatments held throughout the entire summer. The split application of the fierce held out similarly throughout the entire summer. And other than the late fall application of metribuzin, we had pretty good control. Ralston, lower weed numbers, but still good enough to run the analysis, and very similar results to Lind the previous year. And Ralston was probably our driest site of the three sites we have here. But you can see that we're maintaining good Russian thistle control with many of our treatments, particularly the Spartan charge and the split application of fierce. And then if we look at the entire summer, that still holds in there. Our metribuzin starting to slip. You remember in 2020, we actually got some nice May and June rain other than this year. And so we had a bit more of a flush in June than we had in the other sites. And there's where we see kind of the metribuzin and the checks falling down. But again, these soil applications seem to really help with Russian thistle control. As I said, we collected some data on some other weeds, tumble mustard and prickly lettuce. And what we found is while Spartan charge looked really good on the Russian thistle, not so good on the tumble mustard, although the other products worked well, and not so good on the prickly lettuce. And our late fall application of metribuzin also tended to fall apart with our prickly lettuce. So conclusions from this study are that soil applied residual herbicides may be used to manage glyphosate resistant Russian thistle and fallow. They will also reduce the risk of glyphosate resistance. It should hopefully mean that you don't have to apply glyphosate as many times in your fallow and reduce that selection pressure. So this is what they're doing with glyphosate resistant kochia in the Great Plains. They're using both soil applied herbicides and post-emergence herbicides to keep the weed under control. And I think that's what we're going to need to do here in the Pacific Northwest. And across locations, the greatest control of Russian thistle is achieved with sulfentrazone, carfentrazone, so Spartan charge, applied late winter or split applied, or the fierce split applied. We didn't see any injury in the subsequent wheat crop, so it was obviously breaking down. Now, we didn't have a year like we're having now, so this, as dry as we are, this may be more of a problem for particularly Spartan Charge, perhaps, or even the sulfentrazone, the fierce. But in our 3 years of we conducted, we did not see any wheat injury in the subsequent crop. And tumble muster and prickly dust control is poor with the Spartan Charge. It just doesn't control those weeds like it does Russian thistle. I want to talk about a couple more treatments here in the next few minutes I have left. Downy brome and Italian ryegrass are two annual grass weeds that are becoming quite problematic. Italian ryegrass, I mentioned earlier, we have a lot of herbicide resistance in it, and we're developing a lot of resistance in Downy brome. Down in the Walla Walla area, most of the group 2 herbicides no longer work on Downy brome down there. They're ineffective, and we're seeing that spread beyond that area. So the things that we've relied on in the past for Downy brome control are losing their effectiveness. So we want to look if we could find some ways to supplement the control we're getting. So back in 2012, Norsworthy et al. Published a article in Weed Science Journal on best management practices, and the number 6 of the 12 best management practices was use multiple effective mechanisms of action against the most troublesome weeds and those prone to herbicide resistance. So they're saying you should always use more than one mechanism of action. Don't rely on a single one. Unfortunately, we haven't heeded that advice very well. We've tended to rely on a single mechanism of action. And so this study was to see whether bringing in multiple mechanisms of action could help. Now, a lot of people don't know what mechanism of action their herbicides are in. Again, on the Small Grains website, if you go to the weed resources page and overview, you'll see some different tools. And there's a pulse crop mode of action tool and a wheat mode of action tool. And you can go in there and it will tell you what mechanism of action or mode of action the different herbicides are in, and what other herbicides have that same mechanism of action, so you can avoid using herbicides with the same mechanism of action. So in this trial, we put out trialate Avadex as Avadex MicroActiv. That's the granular formulation. Fargo is their liquid formulation. We put out 15 pounds per acre. We had pyroxasulfone, which is Zidua, at 1.5 ounces. Now that was a little, it's not the highest rate, but we wanted to put out a rate that we might be able to see some differences. We had metribuzin added to that at 2 ounces, Beyond at 5, and PowerFlex at 2, PowerFlex HL. We applied the trialate or the Avadex pre-plant. The incorporation was with a high disturbance horse drill. Otherwise, it was no-till. Pre-emergence, the Zidua was applied pre-emergence, and then the post-emergence treatments, imazamox and pyroxasulfone. So Beyond and PowerFlex. The site was near Dixie, Washington, and we did a randomized complete block design. And here's the results on Italian ryegrass. So without Avadex and with Avadex. So without Avadex, none of our treatments effectively controlled Italian ryegrass. No single ingredient or even the Zidua Sencor provided us with commercially acceptable control. The Zidua Sencor was the best, but it wasn't commercially acceptable. Now, maybe if we'd used 2 ounces, we would have bumped that up to a commercially acceptable amount. But you can see that our group 2s just aren't cutting it. You know, Beyond and PowerFlex, neither one of them are providing control like they used to on this. With Avadex, however, by itself, Avadex wasn't good enough. But when we combined Avadex with the Zidua or the Zidua Sencor, we got pretty decent control of Italian ryegrass. The other things didn't really improve it much amongst Avadex alone, but combined, so we had here 2 different mechanisms of action, and here 3 different mechanisms of action working to control the Downy or the Italian ryegrass. Here are the results with Downy Brome, somewhat similar. Again, without Avadex, none of them providing acceptable control of Downy Brome, but with Avadex by itself, again, really not acceptable. But Avadex with Zidua or Zidua Sencor, or now even with Beyond applied in the fall, we were getting good control, or PowerFlex in the fall. These all were doing okay when we had Avadex in there. So I think the lesson is we need to try to get more mechanisms of action working. It would have been nice to do that before we developed resistance to some of these things, but now it's almost imperative that we don't rely on a single mechanism of action to control these troublesome weeds. We also saw quite a yield increase where we used Zidua Avadex and used multiple mechanisms of action compared to where we didn't. So in conclusion, our best telling ryegrass control is when we use both Avadex and Zidua, or Avadex followed by Zidua plus metribuzin. And then the best Downy Brome control treatments were with triolate and those other treatments. So Zidua, Beyond, or PowerFlex fall applied. And so combining two or more mechanisms of action resulted in greatest control. And one more topic I want to talk about, I know I'm getting close on time, is COAXium wheat production system. This is a system, these are some pictures taken looking at rye in Pullman. Highly effective on rye. Similar results when we did a study near Ritzville on Downy Brome. It really does a super job. My concern is that if we rely solely on aggressor herbicide and COAXium wheat, we know the ACCase inhibitors, we develop resistance to them quickly. And if we rely solely on this product, we will develop resistance very quickly to it. So I'm suggesting that you only use the COAXium system if you're going to use other herbicides with it. Put a pre-emergence down, Axiom, Zidua, Anthem Flex, maybe one of these others, and then come back with the aggressor post-emergence, or follow with some of the other ones. But we can no longer rely on just our post-emergence herbicides for Downy Brome control. We're going to have to start mixing in some pre-emergence control into our systems. Don't use the COAXium system more than once every 3 years. They say you can use it 2 times in a row and then you should switch. I'd say if you're in a 3-year crop rotation, so you have crop rotations working for you, you can do it every 3 years. So in wheat fallow, that's every other wheat crop. But in a 3-year rotation, that you, as long as you stay away from using Assure 2, which is the exact same active ingredient as an aggressor, you can get by with this. So you might rotate it with a clear field production system or substitute some different crops into your systems. And this is the slide the WSDA tells me I need to show you. I may have talked about something that's not labeled. I can talk about it, you can't do it. That's what this slide basically says. So with that, if there's time, Lori, I'll take questions.
No, we have plenty of time. So if anybody would like to ask a question, feel free to unmute yourself and ask away.
And I'll get rid of my stop sharing so we can see you. All right.
I guess I'll start us off. This is Ryan Poe, Drew, and I apologize. I have the time wrong, so I missed the beginning part of your talk. But just this, I guess, tail end of this, we've tried some of the Battalion Pro, the biologic for Downy Brome, and actually used it years ago and thought we'd seen pretty good results, but then labeling issues, we couldn't get it. Do you have any trials that kind of are working on if that's an effective control for Downy Brome?
You know, I don't. I believe Aaron Esser has put it out somewhere up around Davenport near the Wilkie farm. I have not done it. I've my knowledge of it is that in the Petri dish, it works really good. Out in nature, it's a little less certain. So, you know, applying an organism to a soil that's already teeming with organisms and it's got to battle with them to survive. And I've heard good reports on it, and I've heard not so good reports. And I haven't seen much data, at least data that's statistically, scientifically backed up and analyzed one way or the other. So a lot of the information is anecdotal. People put it out. It's often not replicated, so you don't know, it's not done in a scientific manner, so it's really hard to know. So maybe it's something I need to work on, but I haven't done so. But as I said, I believe Aaron Esser has put it out. My understanding is you have to wait 2 or 3 years for it to really kick in. So
that's what I guess, just, from, our on-farm trial, like you say, the first time back in crop, like, oh, this didn't do anything. But the next time back in, we saw a huge difference. But anyways, like you say, that's what, we have our experiences, but then I talked to another guy who says that it didn't do anything on their farm. So anyways, and I know timing and, and so many environmental factors, but anyways, I just was curious if there was any trials kind of going on, but like you say, I guess I'll try to follow up with Aaron a little bit.
Yeah, check with Aaron, and it might be, I have never worked with it. I worked with guys like Frank Young, now retired, Alex Ogg, now retired, who worked with Ann Kennedy way back when, and I think they even tried it. Phil Stallman, who was at Kansas State, tried it, and none of them found it to work in the field.
All right, thank you for that. We have a question in the chat box from Scott Williams. Does COAXium have any carryover residual?
COAXium does not. It's pretty much a post-emergence. There's really no soil carryover, so it's not going to exert selection pressure except at the time that you apply it. So yeah, your rotation restrictions, it's gone. It's not going to, if you get subsequent flushes, they'll come up. The thing, a lot of growers like to wait and get all the weeds up and then spray them to try to kill them all. The research would suggest it's the weeds that come up in the fall that are most competitive and produce the most seed, and that these herbicides often work better in the fall than they do in the spring. So I often tell growers, kill the stuff that comes up in the fall, kill it as early as possible, and the later stuff, yes, they'll produce some seed, and but it probably won't take much off on your yield, and your herbicide will work a lot better. It tends to be more consistent in the fall applications than with spring applications.
Okay, any other questions for Drew?
Appreciate your attention. If you have any questions.
I have a question.
Oh, okay. Go ahead.
It's Harold Cochran. When do you apply the Avadex? Is that fall?
Avadex is,
you were talking about?
Avadex is applied pre-plant because their label actually requires mechanical incorporation. We wanted to try it at that Dixie site, no-till, but we did use a high disturbance horse drill, so we were hoping we were getting some incorporation there. We also had excellent rainfall after we applied it, so that helped us. But that's applied pre-plant and should be incorporated to some degree. The pyroxasulfone, the Zidua, can be applied pre-plant, pre-emergence, delayed pre-emergence, and even early post-emergence, depending on when you want to put it down. The rates, you can't put the full load down prior to emergence. So once it's up, then you can put a little more on than you can pre. They're afraid of a little crop injury. So we like to see the highest rate possible used, because we're kind of on the margin of getting good control with the lower rates. So if you do put it on pre-plant, it's usually a good idea to come back early post and put on the rest. Anthem Flex allows you to put it all on pre. they're taking a little more gamble that you won't get the injury.
So can you mix Zidua with Avadex, put them on down together pre-plant?
Yes, you could. You have to be careful though, because what you don't want is I need to change that because you're going to have to incorporate. You don't want to get the pyroxasulfone in contact with the seedling, with the little shoots. It'll nub it up. So it may not be a good idea now that I think about it, because you want to have I don't think you want to incorporate the Zidua, and you do the Avadex. So I would put the Avadex down, incorporate it, and come back with the Zidua.
Thank you for clarifying that.
Okay, you're welcome.
Drew, I got one more follow-up question. So aggressor, from my understanding, is Quizaflop, which we've also used, I guess, in our winter canola. I mean, do you see issues there? I mean, obviously, if we're using it, I mean, does that just progress the resistance if we're obviously using it? Well, and I can't remember now if we use that in winter peas as well, but
Yeah. That is a concern. So in areas where pulse crops have been grown and now canola, Assure 2, the active ingredient is Quizalofop, which is the same thing that's in aggressor. So you're really not, introducing a new mechanism of action. You've already started the selection pressure, put on the selection pressure, and so you're going to increase that by bringing in the aggressor.
Oh,
So be careful. I would decide
no,
if
that
you
stuff
grow
is so dangerous.
canola, or
I know it comes out of
or
nowhere.
peas. Oops, let me mute.
Doesn't
this
make a lot of
person.
sense.
If you grow canola or a pulse crop, I think you need to make a decision. Do you want to use Assure 2 in those crops or aggressor in your wheat, and not do both?
perfect. Thanks.
All right, unless there are no other questions, I would ask anyone who's interested in the 2 pesticide credits. We do ask you stay on through the end of the presentation, but go ahead and enter your credit number in the chat box, and we will submit that to WSDA. And Drew, it doesn't look like we have Tim on at the moment.
Okay, let me see if I can get a hold of Tim.
You could check in with him, and if
Yep.
you need to have Steve go first, we can do that.
All right.
So we'll just give it a few minutes to get everyone's pesticide credit number in there, and we'll bring up our next speaker here in just a couple minutes. Kelly Weber, you might still be working on it, but it looks like I might have an incomplete number for you. Oh, there it is. Ryan.
Sorry, I hit the same button before.
Kelly, I didn't catch all that, but I do see your full number in there now, so you should be good.
So do you have my number, Dennis Schraitland?
I believe I saw it in there. Yep. Gotcha.
Okay, thank you.
Steve Van Vliet, do you hear me?
I do hear you.
Okay, we'll have you on deck in case Tim needs to switch times with you.
Okay, not a problem.
Steve, I think we'll go ahead and grant you.
Lori,
Oh, yes.
do I give you the herbicide number orally?
Yep, that'll work. If that,
Lori,
yes.
this is Drew.
Okay.
Just talk to Tim. He says he's coming on right now.
Okay.
So,
Gordon, I'll just go ahead and call you after the session and I'll get your number that way. If that
23278.
23278. We've got you down. Okay. And I see Tim.
Thank you.
Not moving. Tim.
Are we waiting until 10:10 to start?
No, we're ready. I'm just trying to give you host access. Oh, Drew, that's why. You still have. Claim. Host. And make host. Okay, Tim, you should have host access to share your screen when you are ready.
Okay, great. Well, welcome everybody. I will share my screen and we can start the presentation here. Can everybody see the screen?
Yep, looks good.
Okay, great. Well, this morning we're going to talk about biology and management of stripe rust. Stripe rust, as most of you know, has been probably our most problematic disease over the last several, since about 2010 or so. We've been seeing pretty significant stripe rust most years, and so that's why we're talking about this disease this morning. And in the first slide here, you see two different examples of stripe rust. This photo on the left is sort of the classic stripe rust symptom, where you see these long yellow orange kind of stripes in the leaves. If you were to rub your finger across those stripes, you would get what some people call Cheeto dust or spores of the stripe rust fungus coming off on your finger. On the right-hand side here is another example of stripe rust. It can take on different forms. And one of the things I want to emphasize in this photo is that although you see the long stripes here, we also see these black colored spores in there. And we'll talk about those in a minute. One of the things I'd like to do first is remind everybody that we have three different wheat rusts that we deal with. And all three of these rusts can occur in Eastern Washington, although stripe rust is our most common. Each of the rusts has a different temperature optimum for infection and slightly different disease cycles. Stripe rust has the coolest temperature optimum in that range of 50 to 64. And that's why it's our biggest problem, because those are the kinds of conditions that we see most. We really haven't seen much leaf rust over the last several years. It's the intermediate rust, at least in terms of temperature requirements. You can see there at warmer temperatures, 68 to 77. And then stem rust is the warm season rust, the warmest requirements of all, 75 to 86. We often see stem rust come in late in the season. Most of the time, it doesn't do much damage because it comes in so late. Now, other parts of the world, you'll hear these rusts referred to as yellow rust for stripe rust, brown rust for leaf rust, and black rust for stem rust. They all share the requirement for dew or free water on the surface of the plant for spore germination and infection. So that's very important. Even if you have these temperature ranges or these temperature optimum, but you don't have that dew on the plant, infection will not occur. Here are some more examples, different forms of stripe rust. Here in this upper left photo, I showed earlier the sort of the classic stripe, but this is the kind of symptom that you would probably see on a plant that's a little older, maybe post jointing. In the upper left here is the kind of symptom you would see on a seedling, sometimes called a seedling reaction, where you don't get those long stripes. You get the pustule spread over the surface of the plant. When disease is very severe, the stripes sort of blend one into the other, and you just see the entire leaf surface covered. Stripe rust can also infect the heads, and here's an example of stripe rust in the glooms. And then here's that black spore stage that I mentioned earlier, the teliospore or survival structure of the rust. We see these in Washington in some years, but they're not really important under our conditions. And I'll talk about that in a minute. Stripe rust can be very destructive on susceptible varieties. And here are a couple of photographs that I took many years ago. This upper right photo shows a field that's got this yellow cast to it. This is on the Colotes Pasco Highway. And you can see here the entire field is infected. And when this occurs on a susceptible variety early, yield losses can be up to 70 or 80%. So it can be quite destructive. In the lower left, here's a comparison between a resistant variety on the right and a susceptible variety on the left. And you can see the susceptible variety is just essentially covered with stripe rust, whereas the resistant variety still is green. And that is one of our primary control measures for stripe rust, planting a disease-resistant variety. So we called this talk biology and management, and I want to talk about some of the biology here and what we call a disease cycle, or how does this disease work really. For the most part, and you can see here's that photo again of these yellow stripes here in the lower right. This is the stage that we see, and essentially the main stage that's important for us in the inland Pacific Northwest. This stage is technically called a Uridina spore, and it is produced on wheat and it reinfects wheat. Eventually, this spore stage, as temperatures cool and plants senesce, can lead to that black spore stage that I showed you earlier. And this is called the teliospore. It's a survival spore. And this is akin to the black stem rust fungus that I mentioned earlier. The difference is, and this is relatively new information, we've only known for about the last 10 years that the stripe rust fungus acts just like the stem rust fungus does, in the sense that when these black spores germinate, they can infect the barberry plant. And that's what you see in the upper right-hand panel here. This is a barberry leaf. This is actually infected with stem rust, not stripe rust, but it has exactly the same disease cycle. So the stripe rust fungus can alternate between wheat and barberry. Now, the good news is for us in the Pacific Northwest, that does not occur for stripe rust. It strictly stays in this stage here where it infects wheat and wheat and wheat. The barberry is not important. However, for stem rust, barberry is still important. And so I'm gonna digress here for just a minute and talk about the barberry, because in the inland Pacific Northwest, we're one of the few places in the world really where barberry is still found, and it's still important in the disease cycle of stem rust. And with stem rust, when you have barberries around, it provides an overwintering site and allows the disease to begin earlier in the season. And when barberries are present, the fungus undergoes sexual reproduction on the barberry and results in the production of many new races of the pathogen. And that's one of the common features between all of these rust fungi is the production of new races. And it's very important when we think about races and management of stripe rust. We're going to talk about that in just a minute. I just want to finish up here a few comments about barberry. Barberry is still present in our area. We had an eradication program in Eastern Washington that ended in the 1970s, but we still find barberries around. And there's a number of ways that you can recognize it. One of the really common ways is to just scrape the bark back, and you'll find this yellow color underneath. There's thorns on here that typically are found in threes. In the spring, you may see these yellow flowers. Later in the year, you may see these red fruits. And in the upper left and the lower right here is the rust fungus producing spores on barberry leaves, and then on the lower right on barberry fruit. Okay, so if you see these, you realize what they are and what role they have in rust development. So I want to talk about first, what are pathogenic races? Pathogenic races are essentially strains of a pathogen that are able to infect different varieties or individuals of the host species. In our case, we're talking about wheat varieties. And some years back, we were talking about the flu virus and the fact that there are different strains, H1N1, H3N3. If you get a flu vaccination in the year, it's usually a combination of different races or strains of the flu virus that they expect to be important that year. Well, in plants and with the rust fungi and the striped rust fungus in particular, the same kind of thing happens. And so the ability of a particular pathogenic race to infect a variety depends on which resistance genes that variety contains. And this is, you'll hear breeders talk about breeding for disease resistance and which resistance genes they're using. And this corresponds to different, what we call virulence genes in the pathogen. So this is a very specific relationship, and it applies to one type of resistance in particular. And I'm going to mention that in a minute. So back to stripe rust, back to the disease cycle I mentioned earlier, that temperatures in the range of 50 to 64 with 6 hours of dew are optimal for infection of the stripe rust fungus. These temperatures are also good for disease development, but less important than for infection. You know, once a plant is infected, the stripe rust fungus will keep growing until air temperatures get into the low 90 degrees. So it develops just fine at warmer air temperatures, but infection will slow down. Now, one of the critical things for stripe rust in our area, the stripe rust fungus only survives on living wheat plants. And so there has to be a carryover from the current crop to the new crop. And so the infection of susceptible plants in fall is critical for survival of the stripe rust fungus. And that's where some of our predictions come into play. And then following that infection, the rust fungus has to survive the winter on those crop plants. And temperatures from December to February especially are very important for survival. So if we have temperatures in the single digits or below and without snow cover, that's going to be detrimental to survival of the rust fungus. The rust fungus will die, but the wheat plant will continue just fine. Now, if we have snow cover, that insulates everything, and the air temperatures are not as critical for survival of the stripe rust fungus. So what do we know about stripe rust this year? Well, rust intensity last year during summer 2020 was moderate. We had a relatively dry fall. We had normal to late planting and emergence. And that combined with the fall weather resulted in what I called an average risk for rust establishment in the fall. November and December temperatures were favorable for rust survival, and so the rust fungus came through the winter just fine. And Dr. Chen's first forecast that he put out in January was for severe rust. But even though he made that forecast, the conditions since then were not favorable for rust. And we're currently experiencing the least stripe rust we've seen in the last 10 years. And that's largely due to the very dry weather conditions that we've been experiencing. In Pullman, for example, our weather station at the Palouse Conservation Field Station, precipitation for March, April, and May are the lowest on record since 1894. And that moisture is what is critical for the stripe rust fungus to continue to develop. So that low rainfall is certainly not good for the crop, but it does have a suppressing effect on the stripe rust fungus. So we have several control options for stripe rust. Cultural controls include green bridge or volunteer management, and that gets into that fall infection that I was talking about. If we have a lot of volunteer, especially early seeded volunteer or early seeded crops that are susceptible, that can result in early fall infection and good survival of the rust fungus over the winter. If the wheat is being produced under irrigation, it's best to avoid any kind of excessive irrigation because that moisture helps infection by the rust fungus. Our number one recommendation for control is to plant a disease-resistant variety. Preferably, I say here, those with HTAP resistance and 1 to 4. So what does that mean? HTAP resistance refers to high temperature adult plant resistance. And that type of resistance begins to develop in plants as they start to elongate, as the stems start to elongate or jointing begins. That type of resistance is effective against all races of the stripe rust fungus. So as opposed to seedling resistance, where that is very race specific, adult plant resistance is more general and effective against all races. Now, the 1 to 4 here comes into how we rate varieties for resistance. A rating of 1 to 2 is considered resistant, 3 to 4 is moderately resistant, 5 would be moderate, 6 or 7 would be moderately susceptible, and 8 or 9 would be susceptible. And these ratings can be found in the Washington State Crop Improvement Association variety descriptions and their seed buyer's guide. They can also be found on our website, the smallgrains.wsu.edu website under variety selection, and the new variety selection app that has just come out. So the idea here is to plant a resistant variety. And I always recommend that you look at the WSU variety testing data, see which variety performs well in the area where you're planning to grow it, and then pick the most resistant variety that you can. And if you pick a variety that has a rating of 1 to 4, the need for fungicides diminishes greatly. So to the extent that you're not able to plant a resistant variety or something changes, I always encourage people to monitor the rust forecasts. Dr. Chen begins to put out his forecasts in January. I write something on our smallgrains.wsu.edu website following those forecasts to help interpret them. So there's information there. I also encourage you to scout your fields and then spray fungicides when necessary. So when you're scouting a field for rust, you should spray susceptible varieties, those that are rated 5 to 9, especially when 1 to 5% of the plants have active rust. Okay, so we're going to talk about those two things next year. Here is a list of current ratings for winter wheat varieties, stripe rust ratings. You can see we have them broken down into categories: 1 to 2, 3 to 4, 5, 6, 7, 8, and 9. And the varieties in black are soft white, those in red are hard reds, blue are club wheats, and green are hard white wheats. So there's a lot to choose from here in these top two categories. And again, we're really recommending that you pick something that's either resistant or moderately resistant. This will greatly reduce the need for fungicide application. Okay, we have a similar set of ratings for spring wheat varieties. Again, same thing, black is soft white, red is hard red, club hard white, but we also have some Durhams in here in dark blue. It's a little hard to see, but things like WQL here 195 is a Durham wheat. Okay, so again, same thing holds. Pick the most resistant variety that performs well in your area and reduce the need for fungicide application. Okay, next thing is to monitor forecasts and the development of rusts. I already mentioned that. You can follow Dr. Chen's forecasts, spray when necessary, and that is when susceptible varieties have 1 to 5% rust. What does 5% rust look like? Well, here's a scale on the bottom that shows rust severity from 0 to 100%. And infection type on the top, which is another rating of susceptibility of the variety. And so when we're looking at 1 to 5%, we're down here at the bottom. Well, what does that look like on a real leaf? I took these photos to give you an idea. These are flag leaves. And here is 2 to 5%. That's basically one average size pustule there. 5% is 2 to 3 pustules on a leaf. 10% is a few more pustules, and 15% is a few more. So it doesn't take much rust to pass that threshold for 1 to 5%. Okay. So if you pass that threshold, what do you spray? Well, there are lots of alternatives that you have. Some of the most commonly used fungicides are things like propiconazole here. The trade name would be Tilt, but this is off patent, so there are generic products. Tebuconazole or Folacure is another product that's used widely. Things like Preaxor, Quilt XL have multiple modes of action in them. They have 2 modes of action. And so how do you choose? Well, cost obviously is one choice. If you spray something like propiconazole or tebuconazole, you're probably going to get about 3 weeks of activity if you spray it at a full label rate. If you spray a product that has 2 modes of action in it, you're probably going to gain about a week's worth of effectiveness. And so you can decide when you're spraying, how long you have to go in the season and so forth to help decide which product to choose. There is this other product down here, Triva Pro, which has 3 modes of action in it, and it probably gives you another week's worth of protection. Now, one of the very important things to follow here is the growth stage or the harvest restriction on these materials. Things like propiconazole or Preaxor, they have a growth stage cutoff of 10.5 or 10.5.4. Feek's growth stage 10.5 is anthesis. And so when you see anthers pop out of the middle of the head, you have reached Feek's growth stage 10.5. 10.5.4 is actually the end of anthesis. And so when wheat flowers, the anthers start in the middle of the head and then they move to the top and the bottom. And 10.5.4 is when all of the anthers are out. So with a product like Quilt XL, you might have a week or 10 days longer application window than with Preaxor, depending on weather conditions. Things like tebuconazole don't have a growth stage restriction. They have a pre-harvest restriction. So if you're past anthesis and you think you still need to spray a fungicide, something like tebuconazole, as long as you apply it 30 days or more before harvest, you'll be okay. Now, in general, we don't recommend spraying after anthesis because of just diminishing returns in the benefit of the fungicide. So a summary here for management considerations for stripe rust. The first and foremost, plant varieties with effective resistance, preferably high temperature adult plant resistance, and avoid very susceptible varieties. avoid planting very early, reduce the green bridge, reduce volunteer wheat and other grasses, avoid excessive irrigation, avoid over-fertilization, monitor the forecasts, rust development and reaction of known varieties, scout your fields, and then spray when necessary. And usually an earlier spray application is going to be more effective than one that's made later in the season. Okay, so that's the information that I have for you today. The information that we've talked about is available on our wheat and small grains website. I do put out some information on Twitter that's relevant. The stripe rust alerts by Dr. Chen begin in January, and you can sign up for those at striperustalert.wsu.edu. The variety ratings that I mentioned, stripe rust, eye spot, cephalosporium, are in the crop improvement seed buyer's guide, the variety selection tool, and I mentioned the new app that was just released here recently. And I will have this presentation also loaded onto our website. I wanted to mention too, for some of you, that our plant pest diagnostic clinic, has been closed this year because of our diagnostician changed positions. We are in the process of hiring a new diagnostician, and we expect the clinic to reopen this summer. So watch for information on that. And with that, I will take any questions if we have time.
We do have time, so feel free to use the chat box or go ahead and unmute yourself to ask your questions of Tim.
Any questions? I don't see anything in the chat box. Okay.
All right. Well, I work on giving Steve Van Vliet the host access. If you did not get your certification number in the chat box to get the 2 pesticide credits, please go ahead and do that. We will get those submitted.
And I'll just mention that if questions come up later, you know where to reach me. You can find my email, tim.murray@wsu.edu. I'm on Twitter, and you can go through our small grains team website, smallgrains.wsu.edu.
Sounds great. Okay, Steve, looks like you have host access, so feel free to share your screen. We want to thank Steve for filling in at the last minute. We did have a schedule change, so we appreciate him filling in for Dale to talk on Hessian fly.
Okay. Is everything showing up okay, Lori?
It does show, it looks like it's in presentation mode. I don't know if you'd want to change that because we
Yeah.
can just see your next slide.
I think I can just do that. But you can see it okay this way?
That'll work.
Okay. Okay. So anyway, we're going to talk about Hessian fly today. And of course, the king of Hessian fly is Dr. Michael Pumphrys with WSU. He's our spring wheat breeder. And so I'm going to give a lot of information that comes from his program. He's been doing a lot of that work. He also works in cooperation with Laura Levine and Arash Rashed, which is out of the University of Idaho. So when it comes to Hessian fly will pose a serious threat to not only wheat, but to barley and rye. And of course, wheat is that preferred host. Hessian fly is not a problem when it comes to oats. So we're going to talk a little bit about the background, some detecting infection, and some different control practices of that, and what's been done to mitigate the influence of the Hessian fly populations. So if we look back in time and distribution of Hessian fly in Washington state, of course, I don't know if we like that date, what it starts out with, right? That was when we had the last very bad drought, I believe. So in 1977. But anyway, if you look at the circles that have the little white rings around them, of the distribution from back in that time frame, gives you an idea of Hessian fly populations throughout. And of course, we see them here in Whitman County, you see them throughout. And of course, there's more now, and it's throughout the entire state that we see it. And of course, it's throughout the entire nation that Hessian fly is a problem. In the Western states, we usually have about, and we'll get into the generations, but we usually have about around 2 generations up in this area and in the Northern Great Plains. But in the South, they can have 6 generations of Hessian fly a year. So if we look at what Hessian fly can do, we look at here on the Hessian fly resistance and Hessian fly susceptible spring wheat crops. And you can have a massive amount of loss. You can have up to, if you have a high enough population, you can have 90 to 100% loss. Now, typically we get, you know, in that range of if it's susceptible, up to upwards of 70% loss in high populations. So what does a Hessian fly look like? This is what a Hessian fly looks like. You're not going to be seeing them readily flying around because they're only about, well, they're 4 millimeters long when it comes to the adult female, and it's around 2.5 to 3 millimeters long for the adult male. So very, very small. And when you're trying to find them and trying to identify them, so you're not going to see them readily flying around. Here's some different resources as the other presenters have talked about. You can access a lot of this information on our Small Grains website, smallgrains.wsu.edu. And there's some bulletins put out by the variety testing program that we are starting to list Hessian fly, whether it's resistant or susceptible varieties that have been bred for. So in looking at the Hessian fly in its life cycle, we see up on our upper left what the Hessian fly looks like, and then it lays its eggs on the leaf, underneath leaf surface. And you'll see they hatch, they come down, and you have a short range of time if you're going to try to spray an insecticide, because the insecticide is really only going to be effective when it comes to the larvae of that. Now, will some seed treatments help with Hessian fly control? Maybe, you know, for a short period of time and also a limited amount of control. And it would be more effective when it comes to winter wheat than spring wheat. But we see they move down the leaf down, and they move down towards move down the stem into the crown of the plant, and they'll be feeding. They have a rasping nature. What they start rasping on the plant, and they're feeding on the sugars, feeding on the sap exuded from the plant as they feed, causing those tillers and stems to perish. And of course, then they'll go through several instars, which is several stages, and then they'll pupate into what we call a flaxseed stage. It's a puparium, which is whitish at times, and then it turns into, like we call a flaxseed, because it's very, very small and very, very dark. And of course, when you harvest, you're not going to have them going through the combine because they're going to be down in that lower part of the crown. So, Hessian fly biology importance. Talked a tiny bit about it, but the females will hatch usually with moist, moderate weather conditions. They do lay those eggs on the leaves of cereals and other grasses. So thinking about that, you know, what kind of management do you have when it comes to the management within your different farming practices? So wheat is a preferred host that we had mentioned. There's around 2 generations per year, one in the spring and one in the early summer, typically in this area. And we're thinking potentially could there be a 3rd generation? It's really based on, it's going to be climatic based. So each year can be different. As we know, no year is the same. And it's showing the right slide. Is that correct, Lori? The Hessian fly biology and importance.
Correct.
Okay. So the larvae do develop at the base of the plant, and they do survive adverse weather conditions as this puparia that I talked about. That's a hard coating. That's not going to be a time when you're going to be able to influence the control of that with an insecticide. So the larval feeding results in stunning, lodging, reduced yield, and of course, lower grain quality. So those yield losses in susceptible varieties has ranged from 11 to over 40%. It can be a lot more than that if you have really high populations. I've seen areas in some of our trials, actually over in Makanima. This was years ago when I used to be with industry, but we would see complete losses in some of our susceptible varieties in those trials. If we look at Hessian fly, some of the differences, if you look on here on your left-hand side, you can see the resistant line of wheat, and then you can see how it looks in susceptibility. You can see in testing stages when it comes to the different varieties that have been developed for resistance or whether they're susceptible, what the difference is. So you can see some pretty dead looking plots. Uninfested wheat here in D that has uninfested wheat without Hessian fly and Hessian fly infested wheat. So it really reduces the number of tillers with the feeding of the Hessian fly down in the crown and at the base of that plant. You can see here what the larvae are like down there. You can also see a puparium, and these larvae, I mean, they're just little maggots. So if you've seen maggots, that's what they are, but they're very, very dinky. They form this puparium, a rice seed, and if you split it away, you can see what it does, and you can also see here what that damage does and how it influences the stem and the transport capabilities of that plant for any types of nutrient transport. So Hessian fly is definitely increasing in importance. It's damaging infestations occurring more broadly in the recent years. It's also starting to cause a lot of problems in winter wheat. We actually have pockets or areas that we've noticed it. It's been very prevalent in those areas. Excuse me. That's what happens when you have grad students. So make sure that they go away. So so we do have evidence of resistance source, also these resistances dropping down and thus losing resistance in specific varieties. And we'll talk about this in just a little bit. As I just mentioned, infestation is seen on winter wheat, and some of the areas that we've seen some hot pockets on have actually been in the Dayton, Walla Walla area. There's really been some areas when it comes to winter wheat affected by Hessian fly. And that's becoming more obvious now here in the future. So it's kind of this, often we say the silent pest, because we don't see it. We don't really notice it for a while. And then we have these populations, especially on susceptible varieties, that we like, oh my gosh, now we have a problem. When we notice this problem, they're already in the puparia down there, and trying to get control of it is more or less impossible at that period of time. So resistant varieties can limit that your agriculture economic return from $45 to $104. This is work that was done by Dick Smiley. That's in your, when it comes to your spring wheat loss. So on a year like this, pretty much that's everything you had if you have some spring wheat. So this is a graduate student of Mike's, but this is showing you kind of in the background, you can see pockets of what Hessian fly can do in Walla Walla. This was back in 2017. And then we'll go into some of the different information that comes out of him doing his work on the testing. This is what comes from our variety testing program. You can access this information from smallgrains.wsu.edu. And here we see our different released varieties or experimental lines when it comes to resistance. When it comes to Tico, Seahawk, Louise, Ryan, Witt, all these are resistance. That has been a primary focus of Mike's program. And it's very, very important for us to get resistance of this because, like I say, it's a worldwide and nationwide problem. And so it's been an extreme focus to get the resistance in these lines. Yes, are we starting to see some resistance breakdown? Yes, on a couple lines we are seeing this. So this is typically what it showed in our variety testing program when you went online. Now we're starting to put in, and it'll have in Hessian fly resistance in there. Let's see. I don't want to jump too far. Okay. So here's some different lines also in this area. And of course, you're seeing the yield, what the different averages are. This is a way for you guys to know what is happening with all our different lines in the variety testing program. And then soon you will be able to see which lines all have Hessian fly resistance and what the rankings are. And pretty much the rankings, well, we'll get into this really in just a moment. Going through some of the stuff from Walla Walla and looking at that Walla Walla area. And if you have the susceptible lines, you know, if you're planting those, that's when you're going to have such a loss when it comes to test weight, to yield, and so forth. And ones that are starting to fall out, we'll get into this, but is your Louise and I believe Diva that are starting to lose when it comes to the resistance for Hessian fly. So as you can see down here, these varieties that are susceptible, and I believe Saltese is a susceptible line to Hessian fly. Several of the Syngenta lines that were developed are. And of course, Mike knows all these different ones. I haven't looked at all the different wheat lines for a few years now. Another one comes to yield loss. If you see the ones that are very, very resistant, these are level 1 when it comes to resistance. So if you're looking at Ryan and Seahawk, Tico, these are the ones that are the most resistant to Hessian fly. And of course, those are the ones that are going to be outstanding in yield. And you can even see it when it comes to the test weight on those. So we'll start to lose test weight and we'll start to lose yield when it comes to susceptibility on those lines. And of course, then your lodging is also going to be important when it comes to some of that. If we get lodging factors when it comes to the Hessian fly on that. But here's shows the different 2-year and 3-year averages on that coming from the variety program. This is out of the University of Idaho, but you can see the ones that have excellent Hessian fly resistance, such as Ryan and Tico and Seahawk, and so forth. And probably this 8277 has it too, that they're the highest there on seed yield and within the 3-year average. So what do we need to do? Really, the only way to solve this problem at this point in time are resistant varieties. So that the resistant varieties definitely are going to provide us the most effective means of control. It's very important to use multiple genes for resistance to avoid that breakdown. So we're looking at multiple genes, incorporating those. Mike's program looking at incorporating those into the plant so that we have additional resistance. Just like we do when it comes to resistance to the different diseases, being able to target multiple genes for resistance is the best. Even when, like Tim talking about stripe rust, high temperature adult plant resistance, but you have seedling resistance and you have adult plant resistance, you know, getting both of those in there. It's a little bit different mechanism, but the same idea is true. So what also do we need to do? Rotation and other cultural practices are extremely valuable. You know, not just going with our rotations that we've done all the time. You know, it was typically where we always went from winter wheat to a spring wheat, you know, and then into our pulse crop, especially here in the Palouse. But it was even that way if it comes to some of our areas in the summer fallow, you know, it's wheat after wheat after wheat, and then just a summer fallow bit in between there. And if we aren't maintaining the control of our volunteer cereals and our different grassy crops, we can have some serious problems with Hessian fly. And it's even now, especially when it comes to winter wheat. So we're not avoid of problems if we're just going with, you know, planting winter wheat and then going through a fallow period and then going back into winter wheat, those types of things. Insecticide treatments may help winter wheat more than spring wheat. I mentioned this a bit earlier on that. Some of the rotations and risk: winter wheat, spring wheat, fallow is at a higher risk. Okay, your annual spring wheat, of course, that's going to be very, very high risk. If you're later planting, it's usually a higher risk in the spring, and it's if you're doing it in the fall, because those flies emerge in the late summer and stuff. If you're planting later in the fall, then you could have a lower risk there. But in the spring, that is going to be a problem. So that's why you get the idea of what the difference between that is. Direct seeding typically has a higher risk of that. But if you're doing, like I say, for all of you, whether you're a direct seeder or conventional, you want to use different rotations. It's going to help you tremendously on the management of that, along with you have to plant resistant varieties. Spring wheat adjacent to winter wheat is definitely going to have that higher risk, and poor volunteer control is also, like I just mentioned, is going to be also is a lot higher risk. So we have local populations of Hessian fly are selected. The Hessian fly doesn't travel much, so we're taking local populations to breed for resistance, those types of things. Repeated use of the same resistance gene, though, has led to breakdown that we mentioned. That breakdown is within the Louise and Diva source that I had mentioned earlier. So it's starting to lose its effectiveness. And of course, your winter wheat can be a refuge for Hessian fly. So the breeding lines and varieties are screened under controlling conditions in the laboratory. There's about 16 to 20 plants are tested per genotype using 4 to 5 reps per genotype. That's how Mike does that testing. This is kind of how you see when he does the Hessian fly screening on that. They are screening winter wheats. This is what you're going to see now when you go into the variety testing program, and you'll see this is actually the report. So this is a report that comes out from Clark's program now, variety testing program, that you see whether it's resistant, whether they're susceptible. So you can look at the different ones that you guys have been planting and see whether they're resistant or susceptible. So here's different lines that just enter the WSU variety trial program. Here's their reaction, what kind of infestation rate they have on there, whether it's hard red spring, soft white spring, so forth. Kind of gives you an idea real quick. So this is a sheet directly from a testing there of a location, and you'll see from that the location and what each one of the varieties, how they're reacting, whether we have information on them or whether they are susceptible or resistant. And there's very few that really go into this moderate resistant side of things. It's usually ranked. When it's ranked on that, it's ranked from a 1 to a 9, and I'll get to that here. And it has it. I know I saw it. I'm trying to go back to where. But anyways, if they're ranked from 1 or 2, they're going to be resistant. That's pretty much how they're going to be ranked. They're going to be ranked 1, 2, or they're going to be ranked 8, 9. So you have resistance or you have susceptibility to that. And so when you're planting your spring wheats, definitely get to that. Are there winter wheats that are resistant? There is, and it is, it's almost like it's not showing me a couple of the slides, which is very strange. Huh. Huh. That's weird. It showed me earlier. There is one line that is resistant. Now it's not one that is commercially grown. So they're doing the work on that right now and trying to breed and get more resistance into our winter wheat lines. So we'll go ahead and finish this up. If you guys have any questions, go ahead and ask them at this time. Thank you.
All right, feel free to use the chat box or unmute yourself to ask any questions on any of the topics discussed today. If you have not already done so, please enter your WSDA certification code in the chat box, and we'll be sure to give you your pesticide credits for today. Any questions for our speakers? Or any closing comments from either Drew, Tim, or Steve?
I would just say if anybody has some questions for me, this is Drew. They can contact me at drew.lyon@wsu.edu. I'd be happy to try to answer their questions.
Oh, Tim, you are muted.
Yep, that's the most common phrase of last year. Tim, likewise for me, you can contact me at tim.murray@wsu.edu.
Oh, looks like we've got a couple more numbers in there. We'll save that. And we have recorded this session. And so if it's okay with our friends at WSU, we'll load it to our YouTube channel to watch for playback. But it won't be eligible for the pesticide credits. That did have to be participated live. But the information will be there for anyone who would like to see. So we just want to thank Drew, Tim, and Steve for joining us today. We truly value our relationship with WSU and all they bring to our educational programs. So we hope you found this webinar to be beneficial. And thank you again to our AMO sponsors for making this program possible. We'll plan to see you next year for our Wheat College with guest speaker Peter Johnson when he can join us in person. We'll be in contact with the prize winners, and so our drawing for our Blackstone Grill will go to Gordon Gearing. Congratulations to you. That will be sent or shipped to you just as soon as we order it, and we will send out some new tumblers to Harold Cochran and Scott Williams. So congratulations to everyone on that, and be sure to watch out for information coming on the Tri-State Grain Growers Convention. That will be November 30 through December 3rd at the Davenport Grand in Spokane. So we hope to see everybody there. Safe and happy harvest to everyone. Thanks for joining.
Thanks, Lori.
Thanks, Lori.
Thanks, Lori. Good job.
Thank you. We'll see ya.